Now we can move on to the last presentation. So it will be given by the Dr. Ajinkya Ghagre, who is a PhD student in the Department of Bioengineering at McGill University.
So good afternoon, everyone. So today I'm going to talk about pattern-based contractility screening, which is a technique which allows us to measure contractile forces applied by the cells on the extracellular matrix. So as we all know that the cells apply physical forces and they deform their matrix. This is a fibrillous cell moving through a collagen matrix, deforming the matrix, and this is possible because of the actin and myosin interactions inside the cells. Now, cells they deform the matrix and sense the mechanical properties of the matrix, such as how soft it is or how stiff the matrix is, and based on which they respond biologically, such as increasing the stiffness of the matrix can increase contractility, which can further allow the cells to migrate more towards the stiffer side, or the cells they proliferate more, or stem cells they differentiate into different cell types. Now, all of this is important for growth and development. However, contractility also plays a role in diseases. Like during cancer metastasis, cancer cells they use contractility to pull on collagen and metastasize outwards from a spheroid, or during an asthma attack when there is an abnormal increase in the contractility of smooth muscle cells, which restricts the normal bronchial movement and makes it difficult to breathe for an asthma patient. So all of this motivates us to study how study these cell contractility and ECM interactions, and in order to develop an appropriate therapeutic response. So to study this, first we need to measure cell contractility. Now, traction force microscopy or TFM is like a widely used technique to measure cell contractility. Now, in this technique, first we usually synthesize substrates which are either silicon or polyacrylamide of desired stiffness depending on the cell type which we are using. Now, then we coat these substrates with fluorescent beads and then coat it with collagen or fibronectin for the cell attachment, and we culture cells on these substrates. Now, during a typical traction force microscopy experiment, we first capture the image of the beads with we first capture the image of the beads with the cells on top. We call it a force image. Then we kill the cell, and then we just capture the beads, and we call it a null force image. And then when we compare these two bead images, we get something called as the bead displacement. And from the bead displacement and the material properties that we know, we can calculate strain energy, which is basically the energy applied by the cell to deform the underlying matrix. So this is how we measure contractility using traction force microscopy. Although this technique, it sounds simple, but its experimentation is quite complicated. So now imagine you're doing this experiment for more than 50 to 100 cells. So there is always a problem that you need to capture two images. So you need to go to the first position, capture a bead image, and then come back to the exact same position to capture the null force image, which can be challenging due to microscope drifts or maybe cells moving out of the field sometimes. So which limits the number of cells that you can image. And yeah, and also bead density is a crucial parameter in order when it comes to measuring traction forces. So, and the final thing is that since you're killing the cells in the end, you cannot do any post processing such as immunostaining if you want to compare the contractility with the expression levels of certain proteins. So, uh, to tackle all these issues, we made a technique called pattern-based contractility screening. Now, in pattern-based contractility screening, first using Primo, we print fibronectin patterns on soft silicon substrates. So this is how the fluorescent fibronectin patterns look like on 12 kilopascal silicon substrates. Then we confined fibroblast cells on these patterns. Now, what we saw that the cells, they not only take the shape of the patterns, but they also deform the patterns into these random arbitrary shapes. And this is what really motivated us to calculate forces or calculate the contractility of the cells from these pattern deformations. So basically, in short, we just use this deformed pattern area, and and we know the material properties, and that's how we calculate the strain energy, which is the energy applied by the cells to deform the underlying pattern. So now we have this technique established. So we decided to check the accuracy of this technique. So in order to check the accuracy, we decided to compare it with fraction force microscopy. So we had fiberglass cells on these different shape patterns, but of the same area on 12 kilopascal silicons. And we also had fluorescent beads coated on these substrates. So the beads are not shown here, but there were also TFM beads here. And we measured the traction forces or the strain energy from both only pattern deformations and the bead displacements. So initially we saw a strong correlation between the strain energy calculated from the packs and strain energy calculated from the TFM, irrespective of the cell shape. So we concluded that the packs accurately measure cell contractility across diverse pattern shape. Then we went to challenge packs with different stiffness substrates. So we made the same square patterns of the same area on silicon substrates with different stiffness. And as you can see from pattern deformation itself, that on the stiffest substrates, you have the least pattern deformation. And on the softest substrate, you have like the highest pattern deformation. When we calculated the strain energy, we saw that the strain energy calculated from the TFM correlates with the strain energy calculated from packs. So in conclusion, the packs can also resolve cell contractility as a function of substrate stiffness. So then we challenged packs with some time-dependent experiments. So this is a fibroblast cell on a square pattern of the same area imaged for 18 hours. And by the end of the experiment, we added cytochalasin D to depolymerize actin and remove contractility. And then we measured contractility or the strain energy with TFM and the packs. And we saw that both of these techniques correlated strongly for the time-dependent data. And as soon as we add cyto D for both of these techniques, the contractility decreases with time. When you plot this data together, we can see that the strain energy from packs strongly correlates with the strain energy from TFM. So next, since we had until now we had established the accuracy of this technique, now we decided to move on to studying different cell types to measure contractility, or use different cell types to measure contractility using packs. So we decided to go with breast cancer cells. So we chose MCF7 cells and MDMB231 cells. Breast cancer cells, both are cancer cells, but MDMB231 are highly aggressive cancer cell line compared to MCF7 cells. And we expected that MDMB231 would apply higher contractile forces than MCF7 cells. So we did a similar experiment where we printed square patterns and we confined the two cancer cell lines. And as you can see clearly from the pattern deformations that the MDMB231 cells, they applied higher forces, or they deformed the pattern more compared to MCF7 cells. Yeah, as you can see from the strain energy data. And with packs, we were able to show that the metastatic breast cancer cells, which are MDMB231s, they are more contractile than the benign cancer cells. So in order to explore the full potential of this technique, we decided to print a large amount of patterns in a small area. So here we are printing like 10,000 fibronectin patterns on square patterns of the same area on a 10 kilopascal PDMS substrates. So you can imagine this is a 10 mm by 10 mm square. So it can fit one well of a 24 well plate. And then we confined MCF7 cells on top of these patterns, and we were able to confine approximately 2,000 cells on each patterns. So these are single cells. And then we tried to measure the strain energy. So this is just a histogram of the contractility or the strain energy of the MCF7 cells. And as you can see, the full potential of this technique, that you can do a high throughput screening of a single cell type using this technique in just a single experiment. So in summary, so packs can accurately measure cell contractility irrespective of substrate stiffness or pattern shapes, as we proved by comparing it with TFM. Then it's easier to image because you don't require any null force image. You just have to capture a single image of a deformed pattern. Then it has a potential for high throughput measurements to do more than 1,000 cells at once. And it allows for post processing as you don't have to kill the cells. So you can do some immunostaining at the end of the experiment. So these are just fibroblast cells stained with phalloidin to show actin and DAPI to show the nucleus. And so what scientific questions can you answer with this technique? So basically, if you just want to know what's the contractility of the cells, and so you can just print patterns, seed cell, fluorescent patterns, seed cells on top, cells will deform the patterns, and you can just identify what's the contractility of the cells that you want to work with. Then if you want to study the effect of drugs on cell contractility, you can have cells on patterns, you can treat them with a bunch of drugs, and you can see how the contractility is changing with time. You can also look at the effect of substrate stiffness by printing on different stiffness patterns. You can also look at different proteins involved in making cells more or less contractile. And finally, you can also use this technique to measure contractility for a very long time, which I'm currently using for my stem cells to measure their differentiation for a longer time. So I would like to thank my lab members who really helped me a lot with this project. Sorry for the bioarchive link, but this paper is published in ACS Applied Materials, so you can go through the paper as well. And finally, I would like to thank Alvéole for this opportunity to let me present in the symposium, and also for the fantastic instrument Primo, without which I couldn't have done this. So thank you.
Thank you very much. Oh, there is an echo. Okay, it's okay. So there is no question for the moment on the chat. So I have questions. So to optimize a bit the pattern, did you struggle to optimize a bit the size of the pattern? Because I think that to have single cells, I mean, my question is mostly for single cells. You have to have a precise size, so not too small, in order not to have the cell not spread and not too big, otherwise it will not cover the whole pattern. Was it easy to optimize that?
No, it was not easy to optimize it. So I did try a bunch of different sizes for the patterns. So since I was working with fibroblasts, so around 1600 micrometer square was to 2000 micrometer square was like an optimum for fibroblast cells. If you go more than that, then you see a small group of cells or doublets, which creates an issue. And I was just looking at the single cells. And if you go for in between different cell types, it's different. For example, sometimes I work with MSCs. So even like 1500 or 1600 micrometer square is like a bigger area for them because they are overall their size is smaller. And if you work with something like human dermal fibroblasts, which are huge, so even like 2000 microns is nothing for them. So yeah, so you have to optimize depending on the cell type.
Okay. So we have a question from Kazi. So very nice presentation. How do you apply PLL to the PDMS surface, as normally PLL does not adsorb to untreated PDMS?
Oh, so actually, so it works for us because we were following the exact protocol, which from the alveoli, and we did just the PLL treatment for 1 hour on our PDMS substrates. So it did work for us. It's been working for us really fine.
Okay, and we have two questions from Benoît Ladou. So the first is, what kind of PDMS is it? And the second one is, do forces change with the number of corners within the patterns, between triangle, square, pentagon, etc.?
So for the first question, so we're using the NuSil 800 PDMS, and for do the forces change? Yeah, so they do change as you increase the number of corners, because if you have no corners, if it's a circle, then the forces are like arbitrary. If you like, you cannot predict where the cell is going to apply forces. But if you have like these vertices, then you really constrict the amount of force the cell is applying on the corners or the spots, and you can see then overall increase in contractility as well.
Okay. Okay, well, thank you very much.